Thanks, Anchi. 20 minutes? I think I have 45 minutes, right? Yeah. So yeah, I'm quite daunted, I have to say, because I'm a statistician. I just do a tiny bit of stuff with deep learning, but I thought that this is an interesting and important thing to pick up. So my plan is to first, it's a Mac. Yeah, there we go. To first comment on these fundamental differences between, really not about deep learning specifically, but the fundamental differences between statistics and machine learning. Talk about the sort of unique issue that confounds play in some approaches that have been proposed to deal with them. And then one idea, which I think, the next two things are things I think kind of bridge the two topics. So conformal inference is an idea that's come out of statistics specifically for machine learning cases where you don't have a probabilistic model, but you still want to get probabilistic assurances. And then I'll end with just sort of a showcase of some work from my group showing sort of how we built some fancy statistical models, but they could have existed without some important deep learning methods. So really the core goal of statistics is to try to make inference on a population. So you know, here's a very cartoony example, but you might say, okay, if I can randomly pluck a hair from everyone and then consider the distribution of hair lengths on men versus the distribution on women, a classical statistical question is, can I infer whether the means of the population from which the data were drawn are different? That's what you're doing when you're doing a t-test. You're trying to make a decision about an infinite population. And we do that now. I've tried to illustrate the importance of the difference between a sampling distribution versus the population distribution. Do I have a laser? Yeah. So here, this is the population distribution, but of course what matters when we're making statistical inference is the certainty of the measures we have about the population. So of course the sample mean is narrower than the population, and I've here illustrated the fact that maybe we have more samples from men, and so that's going to be a narrower distribution. We have more information. And of course, if I wanted to test this hypothesis, is the men's hair mean different from the women's hair mean versus so one-sided or two-sided, I would use these sampling distributions to decide. And of course, just visually, we can see that, yes, that it's very likely that any samples you draw, you conclude that these two populations are different. So now, what's the difference in machine learning? Well, machine learning is to actually make decisions about individuals, not the population from which they're drawn. So now I'm trying to say, can I use, for example, hair length to predict gender? Same population, laser. Same population distribution, can I decide? Well, yeah, you could actually use a little threshold rule, but of course, you would be wrong for all the women that have small hair length and wrong for all the men that have long hair length. But that's all you can do in this 1D situation. But this is just illustrating the fundamental approach. I'm not trying to make inference on a population. I'm trying to see what can I do on a subject by subject or unit by unit basis. But of course, just as a footnote, you know, the key to machine learning is that we don't just have one variable. We often have many variables. And just as a cartoon, you can say, well, what if I also knew the number of caps that you wear, say, baseball caps or something? So now, even though these two distributions still have a lot of overlap, I might have a decent job of predicting a new sample, much better than if I just only observed one or the other. So this is a key fundamental idea to machine learning, and of course that deep learning is as good at dealing with these high dimensions, but it is the high dimensional data that give us a chance of making these very challenging decisions. Because in general, you can see that just trying to decide if I only had this data here, I would be really tough. And this is another illustration that in general, it is harder to do prediction than to do inference on a population, right? It's easier to learn, do these two populations have the same mean versus are they actually, can I do a subject by subject prediction because I'm just limited. With enough data, I will always be able to perfectly reject the null hypothesis. With enough data, I will still be wrong on these subjects and wrong on those subjects if I only had one-dimensional data. Okay, so that's the way I would usually like to start talking about machine learning versus statistics. So now what I want to, and then also I wanted to point you to, there's been a number of really great papers that Danilo Bisdock has written, and where he's gone through these two different perspectives. So there was a famous paper written by Leo Breiman, statistician, about these two perspectives on sort of statistics versus machine learning. And there are sort of a number of sort of fundamental differences, where in statistics we have to start with a model, that we have to assume that's true, whereas in machine learning we're just trying to learn from the data. The truth is in the data somehow. We often have way more, the fundamental assumption of statistical tools is usually that we have way more observations than variables, and the converse is usually true in machine learning. We always have to make some sort of assumptions in statistical methods, and of course with machine learning we try to make as few as possible. And then there's just a fundamental paradigm difference of, are we trying to start with some principles and sort of reason to a specific conclusion, or are we just trying to look at data and then eventually sort of learn something about the world? That's this inductive versus deductive. So I think I don't need to go through all these, but it's just two different perspectives on how to treat data, and really they're for asking different questions. You know, if you're trying to infer upon, you know, is there an effect of this drug in the population? Well, that's a statistics question. If you're trying to learn about the sort of the way in which a drug moves through the body by measuring hundreds of variables, that sounds more like a machine learning question. And there's no one, for example, hypothesis that you test in that setting. Of course, there is not just a black and white between statistics and machine learning. There is really, I'd say, a gradient of different tools. So classic tool of statistics is linear regression. Linear regression is basically the general case of almost every tool, t-test, correlation that you might do. And then here at the other end, we have deep neural networks, which are super, super complex, but of course also hard to interpret. So this is a nice, again, this is something from another paper of Danilo's, trying to sort of arrange these, just saying like when you need more flexibility, you have to reach to these kind of tools over here, but you do suffer in terms of sort of interpretability and transparency. And it's not to say that there is not a lot of work to help you understand what deep neural network models are doing, and I'm sure Paul talked about that, but just that it's a lot of work compared to when you have a simple linear model. Okay, so the one thing I thought I would talk about next was confounds, because that's something that comes up a lot, and people ask me, what should I do, you know, when it comes to confounds? And the work that I thought was really neat to illustrate this is some work that was trying to use deep learning to deal with confounds in the context of genetics. So not imaging, but I still think it's a useful setting. So the typical way in a linear model, I mean the way we deal with confounds in a linear model, is that we have some effect of interest. Say, gee, this could be the response, or it could be age or IQ, what have you. And G is going to be some predictor. So this is our predictor effect. It could be patients, controls. It could be, if this is brain, this could be age, what have you. But then we also have confounds. Now, like oftentimes age is a confound. There's sight. There's SES, there's no end of things. There could be a lot of things about the acquisition. Maybe you measure how much the subject moves. That might be an important confound. Okay, but the problem is, how do we know what should go into that X variable? The problem is that, actually, can I use a mouse? I feel like that'd be better, and everyone can see that. I guess I can't use a mouse, that's right. The problem is, well, should it be sex and age squared? Should I do the sex, age squared interaction even? What if there are many highly nonlinear? This is a huge modeling decision. And let's be honest, we always are making modeling decisions, and you make modeling decisions there. But oftentimes there are many, many confounds, and it's not realistic to think that you really are capturing the full space of all the possible confounds. So there are some solutions out there. You may have heard of regression trees, or Bayesian adaptive regression trees. That's one tool that's trying to come up with flexible methods. But this paper interestingly said, well, wait a minute, why don't we put a deep neural network to work on that problem? So what did they do? So they're working in the context of genetics, and so for them, they just sort of if you ignore this is the confounds that they know and understand. These are the genetic PCA components. They're always going to be in there. They actually are okay with those having a linear effect, but they're worried about all the other confounds that could be there. So what they're going to do is they're going to have the effect of interest, the confounds they know and understand well, and then they're going to train a neural network on the confounds to explain as much variability as possible in the response. And then, once that's been trained, they're going to then ask linear regression to estimate what is the importance for this particular outcome. So again, in their setting, they'll actually be using lots of different genes, but they get to train this, they can train this once. So let's see what they did. I don't think I need to go through this, but just to say that the key thing is that they're going to learn this once. So without any genes, without having a particular gene in the model, because we can assume in this setting that the genes do not explain much variance, so we fit a model without this term here, and that's when they train their deep neural network. One thing I really like about this paper is I feel like they gave one of the clearest expositions of a fully connected multi-layer perceptron model, and actually sort of just fully, explicitly write it down in math, not just with pictures of what the model is. So we have an input, we have sort of an intercept that's coming around there, and then we have all these successive layers, and again, they're all fully connected, and you can see the bias terms and the weights terms there. So it's pretty vanilla, but it's very clearly sort of laid out there. So what they point out is that, so first they did some simulations to show that actually, so if I induce some interactions, but then fit a standard model, Bolt LMM is one of the standard models for fitting the genetics data, they show that they can have better power and better false positive control by using their method. So the blue is the baseline, and they're showing that for these different configurations, so you have different levels of genetic variance and different levels of nuisance variance in each case. Not profound, but there's a difference here, and sometimes it's quite appreciable. That basically, when there is more variance attributable to the nuisance, you can do a lot better by doing this. And then for real data, they show they picked 10 UKB phenotypes to do genetic associations on, and they have these confounds and some more confounds when they looked at this vision glaucoma variable. And so then they looked at how many hits do they get with the standard method versus theirs. And what they find is that they actually do, in general, find more hits when they use their deep null. So in general, that's saying, okay, this is an example of using a deep neural network in the context of a traditional statistical linear model to help us better model, better account for the nuisance variation. My only sort of caveat on this, unfortunately, if you look at in the detail of the paper, of course, they do find some more, but they also miss some that the other ones don't find. So, in fact, if you look carefully, so this is the ones from the standard method, this is the minus log p-values from the standard method, minus log p-values from their deep null method. You can see that, yes, there are some that they find, but then, of course, there are also some that they miss. So, yeah, so to me, it was not quite clear that this is a total home run, but I thought it was a really interesting way of combining deep learning and traditional linear model. And that, yeah, this is another one illustrating, yeah. So win some, lose some. Okay, so what do we do in general with confounds? Is that in general, the one thing you might do, and hopefully it's well known that what you should not do, is apply a confound correction to all the data together, train, test, combined. If you do this, you basically are introducing the possibility that information could leak from your test into your training set. And you can, if you just take a bunch of data and you just say, I'm trying to estimate the mean, look what happens when you center the data, the whole data, and then you try to infer on the mean. You get really horrible false overconfidence. So the next sort of default idea then is to use just the training data and to estimate a model that expresses the influence of the confounds on the outcome, and then use those parameters with the held out information, to make residuals, basically to take care of the deconfounding. But this has a limitation that it can be too aggressive. So if there's a lot of shared information between the confounds and your other explanatory variables, it might give you sort of low power. You may not be able to do a very good job in your predictive exercise. So another piece of work I wanted to highlight was from Gal and Farquhar. A moment of silence since Gal doesn't come to OHPM anymore. Okay. let's see what his work is. So his idea is basically saying, okay, we know there are confounds out there, so let's train this as normal, but we're going to create our train and test sets differently. So the normal sort of k-fold cross-validation is to just, randomly just split up the data, and if you have a confound relationship in the test set, you're going to have it in the train set, you'll have it in the test set. And so their idea was to say, well, why don't you create a test set that you carefully select so that there is no relationship between the outcome and the confound. So you extinguish the relationship in just this test set. So now it's not going to change how your performance in your model is going to do necessarily, but what it will do is give you an unbiased estimate of how would this method perform if there was no confound. So they apply this to both CamCam and UK Biobank data, and they do 4 things, and it's sort of really nicely laid out. So you can see what was my prediction. So we were trying to estimate age, so we wanted this to be small as possible. How was it without confounding, with deconfounding and test data jointly, which is generally a bad idea. The out-of-sample method I mentioned, train a confound correction on the test and then apply to on the training and then apply to the train, and then their proposed method here. And to see how good they're doing, they permute them, and so they show the permuted mean absolute errors. And so what you see is that this here is not that different. You're getting relatively similar things here, but what you do see here is the out-of-sample deconfounding does seem to have now just diminished the effect. We have worse error, we have higher error, and it's closer to the null case. In the UK Biobank, they do this as well, and you see even that the out-of-sample deconfounding became worse than chance, because in this case, the deconfounding, the confounds had some implicit information about age, and so they're basically removing that information. But here, the big story here is that you can, if you either without confounding or well, again, this is the non-recommended to found them both jointly, you will think there's a really strong effect. But in fact, when you look at your confound isolated sample, you see actually it's a rather modest effect, and it's rather close to the, and I think there's one more here on fluid intelligence in CamCam, and this is really interesting, basically saying when you account for when you look in a confound isolated sample, there's almost no effect. So that is saying that it's possible that the confounds are actually driving, you're using information in the confounds to drive this fluid intelligence prediction. So it's not really a solution as much, but it's a diagnostic to see how would I be performing in a confound-free setting, and this is saying that you actually are not really doing very well at all. Okay, and then another idea I want to talk about that has come up from statistics is this idea of conformal inference, and I hope I can do it. I find it's rather hard to explain. I've presented it many times, that I'll try to see if I can do the best job possible. So in normal statistical theory, one of the 2 sort of key tools we have is a confidence interval. So in a confidence interval, you get some data from a normal sample, you estimate the variance, and then we have this way of making a confidence interval. Now, to be clear, that is trying to tell us where do we think the population mean is. And the more subjects we have, the smaller this interval gets. And eventually, asymptotically, it converges to a point, because then we now have no truth. But of course, a prediction interval, which in the machine learning setting is what we care about, we want to predict one new individual. This is a different expression. You have to account for the variance of that one new individual, as well as from your imperfect knowledge about the population mean. So here, if n goes to infinity, we don't get a point. We still get an interval that will capture the uncertainty that's in the population. So yeah, so this illustrates, as I said before, how much harder prediction is than inference. But this whole framework here, of course, critically depends on the normality. This 1.96 will only work and only be well calibrated if the data are coming from a normal distribution. In general machine learning settings, we don't want to assume that, and it may just be a highly implausible assumption. So conformal inference, the idea is to say, well, what do we say about our data? We don't want to make any assumption, but the one assumption that seems pretty safe to make is that all the samples that we have are exchangeable. We always assume independence is in any sort of normal statistical procedure, And that seems like a reasonable thing, that our training data is just like a big bag of data, and it's, one can stand in for another. And what we want to do, what we want to do is get a prediction interval or some sort of prediction set. So this is just a formal statement of what the objective is. We want to find some Cy such that the chance that a prediction, that a new value falls into that is at least 1 minus alpha, 95%. And the neat idea here is that you only need exchangeability. Why is that? Well, basically, if we have exchangeable data, we want to see what's going to happen with new data. Well, we can of course, do the whole train test setting and say, okay, well, why not have one new data set play the role, have one new observation play the role of new data. And so what you do is, and again, there's no normality, you define a non-conformity measure. So what you say is, for each new y that I see, how does it comport with the existing values, the true values? And then you actually, once you have that measure, because once you have any measure, you can actually then say, well, how unlikely or likely is this one observation, and you can create a p-value. But then once you have a p-value, you can make intervals. And let me try to illustrate this more clearly. So let's say that the non-conformity is, again, it can be anything, but a typical one is, how far is the new observation from the average? That would be a typical one, and we do that as an absolute error. So it has to be a badness measure. It has to be a loss function. And now, how do I make a p-value? Well, I just say, how much different how does my measure, non-conformity measure, compare using all the data. So each data observation plays the role of a new observation, and we see how this is going to be n different values, and then this is the one proposed value that we have. So this is the weird thing, is I haven't even said where y new is. I'm just saying this is one potential value. But given that I have my n values, I can then make a distribution and see where a given y will fall. And then when we do that, we can then find an interval or some set, basically saying, well, I'm going to play around and try different p-values, find different values of new y, and find which ones I don't reject. And this kind of is exactly how we invert a statistical test into an interval. We say, which values of the true parameter do we not reject the null hypothesis? And those are the ones that go inside the interval. Which values of the parameter would we reject the value? That's where we would not, that's outside the interval. That's where we would reject the null. But we have not made any assumptions about normality. So let me see if I can try to illustrate it. It's kind of sort of artificial, but one of the simplest machine learning, simplest prediction problems is just saying I have labels but no features. So I just have data, and in this case I can have data. Data that is a bimodal, bounded distribution. And if you just naively try to compute a 95% prediction interval for this observation, for this sample, you will get something that goes from some negative value to up here. And even though I asked it to create a 90% confidence interval, I get almost 100% coverage because the data are highly non-normal. So now what I can do is I can propose a nonconformity measure using the average, and this has now the nominal coverage. So now I'm being less, I just have a smaller interval. Now it turns out it's not perfect, it is showing me negative values which are impossible, so there are some other nonconformity measures that you can try. So one nonconformity measure you can try is how far away am I from the median in quantile space. So if I have an observation that is at the 25th percentile, this value here would be 0.5, because I'm 0.5 minus 0.25 minus 0.5 is that distance. And when you do that, you get a method that obeys the bound of the distribution, and I still have nominal coverage here at 90%, and I have an interval that does not go beyond the range of the data. Now, of course, the one last thing it doesn't do here is it doesn't capture the bimodal structure. So there is something else that you can do. It's a bit hard to explain, but what is the most natural conformity, non-conformity measure? Well, the likelihood. So if you can get a density, an estimate of the distribution of the data itself, and of course, because it's not, so the probability distribution is basically how likely is a given value of data to occur. So we need a non-conformity measure, so we have to make it minus this distribution. But if you use that as your non-conformity measure, then you can get things like this. It doesn't even have to be an interval. So you can just say where are the 90% most likely values for this distribution. So this is just a 1D, no label, label only, no feature example, but it gives you an idea of the flexibility. There's been no normality assumption here. All we've assumed is that we can exchange, that the data are exchangeable, and then we can choose the appropriate non-conformity measure. So example from our work in lesion segmentation for MS is I'll show that in a second, but the motivation is that if we're going to do something for segmentation, well, we need an appropriate non-conformity measure. If we have a mask and we're trying to learn where is, for example, a tumor or lesions, we thought that what makes sense to us is a false negative rate. Well, false negative rate is 1 minus sensitivity, right? So sensitivity is a goodness measure. You have to turn it into a badness measure. So 1 minus sensitivity is a false negative rate. So basically, 1 minus how good am I doing at finding the lesion where there are lesion voxels. And then I just use that as my loss function, as my non-conformity measure in the conformal inference. So here's an example of how we put this to work in some MS lesion work. I'll be saying more about that in a second. And so what we, as with all segmentations using deep neural networks, you get a continuous image out, right? And then you apply some threshold to figure out what values are defined as lesion or not. And so what we are able to do then is to use this conformal approach to see what value of the threshold should I have. So we chose 0.3, or in other words, we wanted no more than 0.3 at false negative rate, or in other words, at least 70% power. So now we have this guarantee on, so here's ground truth, and so we don't get everything, but we get most of these lesions here, and we're guaranteed that over the data set, we have, by appropriate choice of this threshold, we are controlling the false negative rate at 30%, or at least 70% of, we are detecting at least 70% of the things that are there, voxels that are lesions. So that's just one example of how we put it to work in our work. So let me say more about then multiple sclerosis. I'm part of a big project based at the Big Data Institute, this is one area where we've been using both statistics and machine learning. If you're not familiar with multiple sclerosis, of course it's a severe disease. It's actually one of the diseases that depends on MRI. There aren't too many things, you think about this, when people have a diagnosis of Alzheimer's, generally they're not going to get an MRI, maybe just to rule out other things. MRIs are used in research for Alzheimer's, but we don't necessarily use them that much for treatment, whereas people get regular MRI scans to track the disease of multiple sclerosis because it has these lesions, these little marks of inflammation. And the current understanding of the disease is that you have a starting point where you basically have a relapsing remitting state, you are fine, then you have these attacks where you have disability, and then you get better, and then you have attacks, and it generally gets increasing disability, and then at some point you just get changed to just no more relapses, you just get severely more and more impaired. But the thing that is motivating our work was that every sort of few years, the MS people sort of change what these labels are, because really we don't know. You know, first they thought there was something called clinically isolated syndrome, and now they don't think that's a thing anymore. They change what these boundaries are, because all they're basing it on are just their experiences with patients and looking at the scans. So our motivation was, well, can't we use, and the motivation that Y-Novartis set out to work with us, we have all this rich data, can't we use that data itself to discover what is the structure of MS? So this is the KNOW-MS data set that we have prepared in collaboration with them. We have, as you can see, over 30,000 patients, over 200,000 MRI scans, in some cases up to 14 years of follow-up. It's a really, really remarkable data set. And so what the first thing we did is we built a deep learning approach to modeling the lesions. So we did have some lesion masks that were created by contract research organizations, and then we used those to train, but the problem was there were different CROs that did each of the work, and they wanted one consistent way to analyze all this data. And so we did this. But one of the things we immediately noticed is that there's really big differences in the number and the volume of lesions. You know, you can have some small, low ones, but you can also have, you know, intermediate and high. Some are very, even when you have lots, that you can have some that are small and some are bigger. So the first thing we found that we can improve our classification was if, sorry, this is the network we built. I think it's pretty vanilla UNet standard model. And we have a private cloud where we do all this work. We use a lot of augmentation. One of the things challenges are that lesions, even though you know, these are sick patients, they have a lot of lesions. By and large, most of the brain is not lesion, so it's a severe class imbalance problem. So we do a lot of data augmentation, and what we found is that we can improve the performance by segmenting the data into these 4 classes of low number, low volume, high number, high volume, high number, low volume, and low number, high volume. So this is low number, high volume is basically when they all start to get confluent and sort of run together. And so we found we could do better performance when we actually just trained 4 different models, and then we, of course, run everything on these 4 models and then take a consensus vote to decide where there is a lesion or not. And yeah, and so this is something, just a little feature. Come look for us at poster 1886. We have some work on trying to deal with this class imbalance problem. There are a lot of different weighted measures I never was aware of. Tversky's, the modification of dice, and modifications across entropy. We have gone and exhaustively compared all these different types of cost functions as ways to try to deal with the class imbalance problem. So. And we found we can do a lot better by looking for the right cost function. So, a little advertisement there. Okay, so, oh no, there's so many builds. Sorry, I didn't want that. Okay, so our idea was, let's take we know that people are evolving in this disease, but we also know that there's a lot of heterogeneity. And so the question is, can we let the data itself sort of figure out what common states that people are in? They might change, they might be come in at higher advanced states, but can we just let the data itself find the models? This is work led by Habib Ganjgahi, and this is and so the goal is to say, can we find these key dimensions using both the radiological features and the clinical dimensions? And so he proposed using a hidden Markov model to basically consider that there are these hidden states. We don't know what they are, but we know that people may stay in them or they may transition. And at each one of these states, we have these observed variables. These are clinical scores. EDSS is a standard measure of MS. Disability, PSOD is of cognition, 9-hole peg hole test, and walking distance. And we just, based on these measurements, so they're called the emissions of the chain, can we discover what the actual true latent state is? But one thing we ran into right away is that there is a lot of missing data. Not all observations have the imaging data. There are more observations about all these clinicals than the imaging. And some of these things are binary, and these are not part of a standard hidden Markov model, just has a Gaussian model. And the standards one would also assume independence. So Habib came up with a bunch of challenges by basically dealing with the missing data by having a fully specified Bayesian model where he can then just integrate out the missing data. And he dealt with the discreteness by saying, well, why don't we first do a factor analysis model? So we have these 7 measures, but you can see there's a lot of correlation between them. Some of them negatively correlated, but there's a lot of dependence in here. So, and another standard problem with standard HMM is that you have to specify the dimensionality. How many hidden states are there? So using a fully specified Bayesian model, he integrated a factor analysis model into the Markov model. So we use the data itself to find the dimensionality that's there. And the key thing is that by projecting it to a lower dimension, we also are making it more normal. So we now have independent and more continuous measures which are more compatible with the standard HMM model. And again, there are different variants of the HMM, but we wanted to use the variants that allow it to learn the dimensionality of the data and those standard ones, those models that depend on Gauss unity. So this is the data we applied it with. We used 8,000 MS patients, using about 6,000 for discovery and 1,600 for validation. I think the details aren't too important here, but the key things are, what do we fit in? We're basically saying, based on these clinical measures, so basically this disability score, physical, so walking ability, agility with your hands, cognitive, and then are you relapsing right now? Yes, no. And then number of GAD-enhancing lesions, number of T2 lesions, and the normalized brain volume. GAD-enhancing lesions are an indication that there's inflammation happening in the brain right now. They're kind of like a very transient signal, but they're very important to say that there's damage going on right now. T2 is kind of more like a running integral of the damage to the brain. Okay, so the first thing that happens is there's a factor analysis. And so the factor analysis found 4 factors that identified physical disability, combining these measures here, and then diffuse brain damage, which is sort of connecting both the T1 lesion volume and the normalized brain volume, and some of the cognitive and some of the EDSS as well. And then the other 2 have found, basically it's completely just identified relapse as having sort of unique isolated information, and then something that's just about the brain. So basically it's about the lesions. So now we'll fit a Markov model on these 4 dimensions instead of the original 7. And what we find is pretty neat is that we actually find a structure where, find a structure where people basically, there's relatively lower disability and gradually increasing disability as we go along here as it color-coded. So you can see as red as these are the highest end of these scales, this is the most disabled. And so you have this end down here, these are people that are least disabled down here, and then, but what's interesting, they stay in that state. They tend to just go back and forth, and then they may go to one of these 2 states, 4 and 5, where they are having relapses here, or relapses and T1 gadolinium lesions. And so, and then you have these severe states. But what was completely discovered by the model is that you only can get to these severe states by coming through these states here. You cannot, and of course, once you're here, you can go back. So this is relatively healthy, and then you have a relapse, maybe with lesions, and then you come back, or you can go on. But these are basically gatekeeper models. So we can also compose this, yes, we call this early MS, transient, and evolved MS, although the clinicians argue a lot about what we should call this. So you can make metastates, and it's neat. So we actually can, we've basically just asked the data itself to discover this, and we have examples of these people in these different stages, but we can now apply this to new data and have done that and validated that it is also getting similar performance on the held out data. So I'll just skip this. So my main point here was, aside from just advertising what's been done in my group, is that this is a really perfect example of using both deep learning and statistical models hand in hand. We wanted to understand the structure of MS, but we wanted to use information about the fact that it's an evolving disease and people change in the sort of the state of the disease, and they tend to stay in a given state and then they change. That is perfectly captured by the hidden Markov model, a classic statistical model. But it wouldn't have been possible unless we had the radiological features that came from a deep learning model. And that was, wouldn't have been possible without those 2 things together. Right, so I think I'll stop there. So I think it's hopefully made some illustrations on the difference between statistics and machine learning. One is about trying to make calibrated inferences on features of population, and whereas machine learning is trying to just make predictions for individual units, although getting some help from statistics on trying to get some of that calibration as well. And really they just have complementary roles. We needed the ML methods for the features, and ML needs the statistics for the calibration. So I'll stop there. Thanks.
We have time for questions. Any questions? Hey, Tom, that was a great talk. Thanks. I was just wondering, does the factor analysis, or not the factor analysis, the hidden Markov model, do you think that indicates that there's not really a construct of primary progressive MS? Or kind of, how do you think about primary progressive MS in the context of that Markov model?
Yeah, I think, do I have it here? Sorry, we have, here we are. So we have the traditional existing type, sorry I didn't highlight this, primary progressive, secondary progressive, relaxing and remitting. And all I can say is that our model is saying it's more complicated. There's no, like, when you have a relapsing and remitting, yes, mostly you're here, but we have half the people with evolved MS have primary progressive. So we're really, yeah, just trying to say that these existing labels leave a lot to be desired, and we want to try to be able to get to a data-based replacement. But yeah, I mean, I wouldn't want to start a fight with an MS-ologist about this, but we're trying to change the status.
Any other questions? If not, let's thank Tom again.